Thank you and good morning. Today I'll be talking about ways of using DNA for investigation and homicide investigation that go beyond what you're used to from your crime lab. Let me begin with what is DNA and how does it work. I'm not going to talk about data or genotypes or match statistics. I'm just going to show you how it's actually used. So imagine that we have an individual, shown here as a well-known individual, who deposits their DNA. And they deposit their DNA, and essentially after the DNA has been collected and it's been turned into genetic types, it leaves essentially a barcode. It's called a genotype, but we'll just call it a barcode, a genetic barcode. And the concept is that if DNA is deposited twice, maybe as two items of evidence, leaving two barcodes, or as a cheek swab, say as the right barcode and evidence as the left barcode, then we have barcodes left by one person, and the question is, can we then associate these barcodes and determine if they were left by a common person? And the way that's done with modern computing is to take the barcodes, compare them from the items that may have been left on a handgun or a surface or a cheek swab, and produce a match statistic from them. And here, the match statistics that I'm showing, 10 is a big number, so I'm showing it as positive 10. You can also think of 10 as a 1 followed by 10 zeros, which would be 10 billion. So this shows the size or the number of zeros in a match statistic. And if there's an association between barcodes, genetic barcodes left in different places, then we have a concept of DNA, deposit and associate, right? The DNA is deposited by one individual, and then the association between them leads to a number, a match statistic, in this case 10, that says, yes, indeed, that hypothesis that you had as investigators that maybe there was someone in common between these two items that where a barcode, genetic barcode was found, makes sense. There's a match number that's big, say plus 10, across these two barcodes from two different items, and that implies a common individual who has deposited their DNA as determined by this association. So what I'm now going to do is go beyond the confines of what happens in most crime labs across the DNA universe of what's possible. And so if instead of having people looking at simple data signals, at the simplest of barcodes, and trying to make a determination if someone's present or absent from DNA, instead a computer can take a look at damaged barcodes, it can look at combinations of barcodes, and pull out those genetic barcodes and extract each barcode from the evidence in order to make comparisons. And so that's what TrueAllele does, and I'm going to tell you how you can go beyond what you're used to. So imagine that you want to establish an exclusion, right? We have John in the center here, and following that, his red line, he's left a barcode on some item of evidence. And now we have another bloke, Paul, and he's left his barcode, but they're actually different barcodes. Now, when we're analyzing these data that you bring into the laboratory and then can get analyzed by a computer, we don't know what these barcodes are. What if they're different? Well, if the barcodes are different as they are here, what a TrueAllele computer can do that virtually no crime lab can is to give you a number that says to what extent are these two barcodes different. And so if there's not a positive match of plus 10, but a mismatch of negative 10, you can think of it as 1 over 10 billion, then we've shown that these two items, examining their DNA, that DNA was not left by the same person because the barcodes are different. So there's a non-association between those barcodes. We can also look at low-level or degraded DNA. Instead of the usual result that you may see with small amounts of genetic material, or with degraded DNA where the result is inconclusive, a result that you may see a lot from crime lab reports. Instead, shown on the left is a fuzzy barcode. That is, the evidence is the DNA data can't produce all the information that might be present in the individual's DNA who left it. But just because it's fuzzy doesn't mean that it can't be looked at. If you look at an old black and white TV, it's fuzzy, you can still get an image out of it. You don't just throw it out as inconclusive. And so if those green lines converge and fuzzy evidence on the left is shown against the sharp cheek swab on the right, that barcode, as the green lines converge, you can still get a match statistic. Maybe it won't be a 10, maybe it'll only be a 6, like a 1 followed by 6 zeros or a million. But you can still get a match statistic that shows that this fuzzy or degraded DNA evidence, shown on the left, does associate with the DNA from the individual's reference barcode on the right, and there's a common person here who's left that DNA. It doesn't have to be inconclusive, it's just a lower number. What about a mixture? What if you have more than one individual? This is where crime labs start telling you a lot of things are inconclusive, particularly if there's a major contributor. Here we have Paul as a major. Think of Hey Jude, right? You hear Paul singing all the way through, right? And not that much John, a little bit of backup harmony. So in that mixture, there's a lot of one individual, shown larger here on the left, and a little bit less of another individual. But a computer like TrueAllele can separate out those barcodes. It can see there's a lot of one individual, that's the Paul barcode, less of another individual, may only be 20 or 25% of the mixture. Instead of calling it inconclusive, when a comparison is made with John's barcode, there's a match statistic. It can be quite high, say +8, because the computer can separate out the barcodes from the evidence items. So let me give you an example. It's a little more concrete, because you usually don't work with barcodes every day. You work with things more like handguns. So this is a case from a county in Pennsylvania where there's a swab from a handgun. And the lab report, as it usually will say with a 4-person mixture, is that it's inconclusive. No conclusions can be made regarding the persons who were compared with A and B as possible contributors to the mixture. All right? Well, what happened when we took that inconclusive DNA, that is the same data, inconclusive to human review, and then ran it through the computer? The computer was able to separate it into 4 different barcodes, 4 different genetic types. And by unmixing the mixture into the barcodes of what was present, then comparisons can be made. And it's just like old DNA from 15 years ago. If you separate out a complex mixture into 4 different barcodes, they're just like the original DNA we had with single source DNA from one person, just maybe a bit fuzzier. So in this case, comparison was made with 2 people. The first person, A, didn't have the same barcode or genotype as any of the 4 contributors that were found on the handgun. And so he was statistically excluded with a negative dissociation, and the district attorney dropped charges against him. However, another person, person B, did have a barcode that was similar. His DNA was similar to one of the contributors, in this case, the 3rd contributor. That match statistic was 400,000, which has 5 or 6 zeros, 5 zeros after the one's about 500,000. And that person was shown to be present on the handgun, and he pled guilty. And so the result of having a separation of DNA by computer into 4 components, instead of it being inconclusive and then going to court, not having physical evidence, and so on, instead one person goes free, the other person pleads, no expense or hassle of trial. The DNA is doing what it's supposed to be doing, which is resolving disputes about identity from evidence. There's a lot more that can be done with DNA than just seeing if a person has left their DNA on an item of evidence. What can also happen is that you don't even have to know who the people are. Evidence from one item can be compared as DNA evidence with another item, even in the absence of people. because all that's happening is there's a statistical association that's being made between these barcodes. So suppose we have one evidence item shown as the barcode in the middle of Paul and John, because Paul and John left their DNA on one item of evidence. On the right is another mixture of genetic barcodes. This is John and George, and we see from the red lines they contributed their DNA. Well, in fact, by separating out the barcodes, the genetic types that's common to John in both cases can be extracted, compared, and statistically matched. Here a number of +8 is shown. So one item of evidence could be compared with another item of evidence. In other words, you don't need a suspect in order to start dissecting a crime scene. You just need the DNA and computers that can separate out barcodes and make reliable comparisons. So in this case, this supports that number +8 under the green arrows shows that there's an individual, whether we know who he is or not, John, who's contributed his DNA to both items of evidence. So here's an example where that evidence-to-evidence comparison was really helpful in southwestern Pennsylvania. This is a murder that occurred in McKeesport. It was an after-hours club, and evidently people were standing around, and someone pulled out a gun and shot and killed another individual. He fled the crime scene as he was running out of the bar. He threw his gun under a car. He dropped his hat, scaled a fence, and disappeared. So now there are two items of DNA evidence. And this is all that could be presented in court. There may have been other evidence of witnesses who wouldn't come forward, who could tell investigators more about the crime, but it couldn't be used as evidence in court. And the prosecutor did not want to go forward without two items of DNA. And so he had two items of DNA. It turned out that they were both three-person mixtures. So shown on the left is 3 barcodes under the gun. Those are the genetic types that were contributed to the handgun. Shown under the hat are 3 barcodes. Those are the 3 genetic types of the 3 people who left their DNA on the hat. And it turned out that there was a defendant. His name was actually Leland Davis. It wasn't Ringo. But when comparison was made to his genotype, there were very strong matches. So the top barcode in all cases labeled Ringo is matching with a strong statistic a suspect. And so the handgun has one of the 3 genetic barcodes that have been pulled out by a computer, matching Ringo with a number of +9, or about 1 billion, 1 followed by 9 zeros. And Ringo's barcode is matching one of the 3 hat genetic types or barcodes that have been pulled out by the TrueAllele computer. That number is +16, which is 1 followed by 16 zeros, about 10 quadrillion. So there's a very strong match, and so now it's clear there's one individual who's contributed his DNA to both of these items. But the presumed defense would be, sure, you found Ringo, but there's somebody else, maybe Pete Best, some other individual who's actually the true perpetrator, and you can't say anything about those other barcodes, those other genetic types. It's the unknown person who's in here. But by having the computer separate out these barcodes, 1, 2, and 3, and what's shown here next to the gun are the 3 numbers of the barcodes, 1, 2, and 3, and the percentages of their composition in this mixture, how much of each barcode is there, which truly also works out. There's a 17% component, a major 63 component, another smaller 20% component. Across the columns next to the hat, there were similarly 3 individuals who contributed with barcodes, 9%, 9%, and 81%. And when these separated barcodes were compared, as shown in the center of the last column, there's 9 zeros. We're seeing a match to the defendant with a +9. It's about 1 billion, but nowhere else is there a match. So in fact, there wasn't another person, and Mr. Davis was convicted very much on the DNA evidence. What about crime scene investigation? Well, if you have all these barcodes coming from evidence and people of DNA that's been left at a crime scene, you can begin to start pulling it apart. Here, for example, here's a picture where there's a gun with two barcodes, one from John and one from Ringo. On the right is a hat, say, from Ringo and from George. Ringo is matching Ringo through his barcodes. He may not even be known at this point. So we know the hat is matching the gun. And there's innocent Paul in the corner, right? He didn't do anything at all. And he's not associated with this crime. And that would be determined by having negative associations, showing that he's not associated with any item of evidence. This was very helpful in a case that I worked on in Southern California, where there was a gang that was getting increasingly violent and terrorizing a county. It was actually Kern County, where Bakersfield is. And they visited a food mart, a hardware store. They began tying up the victims and beating them, and it was getting very much out of hand, and people were expecting the violence to really accelerate. In this case, this county actually has the TrueAllele system, and they use it for purposes like this, but it hadn't come online yet. And so they asked us to look at all of this data, and we did. So, for example, at the jewelry store in the center, there's DNA left by 2, 3, or 4 people in very low levels on a counter and on a safe. Different items of evidence, and we could see what the associations were even before we received any references of any individuals, to see which crime scenes were associated and who left their DNA on which items. When we actually received the suspect DNA shown at the top, suspect 1, S2, S3, and so on, we were able to completely dissect the crime scene and figure out who left their DNA on which items. And this was very helpful in understanding who had done what, who on the video footage corresponded to the man who left his hand on the counter as a 3-person mixture, by pulling out these barcodes and connecting evidence with individuals. This is a case from Allegheny County where there were spent shotgun shells. Many crime labs will tell you on spent shells nothing can be done. Well, in this case, it was sent to a private lab. Work was done. The lab found it inconclusive for many reasons. It was low-level DNA from spent shells. But what the crime was that, or the hypothesis was that a mentally unbalanced young man had come home and had shot both of his parents in their home as he returned after a 5-year absence. And the DNA mixtures, as shown under the shot shells, were mainly the father, the son, and the mother. And that was determined by having the computer separate out the DNAs of these related people, which most crime labs can't do. They can't handle mixtures of relatives. They can't handle low DNA. They can't handle 3-person mixtures. But it makes no difference if you can separate out the barcodes and then make comparison. Ultimately, the son was compared with the son component, about 30% of the mixture, with a match statistic of plus 12, as 12 zeros after the 1 is a trillion. So in Pennsylvania, so far, Cybergenetics has worked on over 75 cases. In Allegheny County, we've worked on 44 cases. We've had 8 trials. There have actually been 3 DNA exonerations when we showed that there was a negative number and a person was not associated with a crime. If you look at this table, these are the guilty outcomes that I know about so far. There are about 20 of them. There are a number of murders, and most of the results are guilty pleas that don't need to go to trial because the DNA is clear from the evidence. What's also clear, if you look in the second column under evidence, is that these are not the usual body cavities and so on from biological evidence, biological items. They're from guns and hats, fingernails, clothing, doors. So wherever DNA is left, if someone touches a gun and 3 other of his friends touch it, and there's 4 different people on that gun, it can be pried apart by computer to get at those 4 barcodes for comparisons to be made in order to determine a match statistic. Basically, the older methods of human review that have been recently more discredited in the press in states like Texas, the Washington, D.C. lab shut down completely because of misinterpretation of DNA mixtures. Shown in red are some of the reasons. The methods are biased. They have thresholds that throw out most of the data. They can only include. They can't include or exclude. Their statistics just count how much Money was spent on tests, not actual probative match information. So the methods are not terribly objective. They've never been validated. TrueAllele's had over 30 validation studies, 7 of them published in journals. They've been shown to be inaccurate. They've never been subjected to a Frye challenge. It's unclear they would survive one. They can't do item-to-item matches of evidence. They can't handle low-level touch DNA. For mixtures, they can tend to handle 2, maybe 3 people if it's the major contributor, 50% or more, whereas TrueAllele computer, we have a validation that's now going out to 10 unknown contributors. Baltimore's validated with to 6 unknown contributors, and it can handle reliably. It can do 1 to 5%, but usually once you have a minor component that's at least 5%, you'll start getting match results. And I won't have no time today to talk about DNA databases, but as you may know, with the CODIS database, maybe 10% of mixtures, and even then only the major components, can be uploaded. So for homicide investigators, CODIS is not that helpful when you have mixed DNA evidence. There's a whole other story that you can look at online and read about with TrueAllele with a mixture databases. By separating out the DNA barcodes from the mixtures, essentially 100% of the evidence can be uploaded to a DNA database and matched against evidence or against people and used instead of discarded for investigative purposes. So there's more information on our website, www.cybgen.com. Lisa's here today. She can tell you more. How do you get started? Basically, if you have an interesting crime, arrange for your laboratory, state or otherwise, to send us the data. We'll look at it at no cost and let you know what information is there. Thank you very much.